Hello students, in this session of chapter number 11, that is enhancement of food production, we are going to learn regarding the fifth type of biofertilizer, that is fungal biofertilizers. Now fungal biofertilizers were first discovered by the scientist called as Frank in the year 1885. Now these fungal biofertilizers are called as mycorrhiza, and these mycorrhiza form a symbiotic association with rhizomes, then roots of the plants in humid region. That means this mycorrhiza is associated, or it forms symbiotic association with the roots of the plants found in humid region. Now you know that fungus consists of hyphae, then hyphae come together to form mycelium. So the fungal biofertilizers are of two types. The first one is ectomycorrhizae. Now the name itself indicates, ecto means they are present on the surface of the root. Now the mycelium forms a mantle on the roots. Now here in this diagram we can see the root, that is taproot, and this taproot is covered by the mycelium of fungi, and it forms a covering, and it is called as fungal mantle. So this type of biofertilizer or fungal biofertilizer is called as ectomycorrhizae, as it forms a covering or a layer around the root. It is called as ectomycorrhizae. Now this ectomycorrhizae helps in the absorption of water and minerals to the roots. Then some fungal hyphae penetrate the cortex of the root and are present in between the intercellular spaces of the cortical cell, and they form a net-like structure called as Hartig net. That means the hyphae form a network inside the intercellular spaces of the cortex cell, and this network or net-like structure is called as Hartig net. So ectomycorrhizae, which are present on the surface of the root forming fungal mantle, help in the absorption of water and minerals. Then the second type of fungal biofertilizer is called as endomycorrhizae. Now the name indicates, endo means the fungal hyphae grow inside the cortical region of the root. Here is the cortical region of the root. So these fungal hyphae grow in between the intercellular space of cortical cell and form two structures. Now the branched structures, here we can see the branched structures. This branch structure of the hyphae are called as arbuscules. Then they form spherical structures, and these spherical structures are called as vesicles. So as endomycorrhizae forms two structure, that is vesicles and branch structure called as arbuscules. The endomycorrhizae are called as vesicular arbuscular mycorrhizae, and its short form is VAM. So fungal biofertilizers are of two types, that is ectomycorrhizae and endomycorrhizae. So these both the types of fungal biofertilizers help in the absorption of water and minerals to the roots. Now we will see the next topic, that is biofertilizer microorganisms, in which we are going to learn regarding different microorganisms which help in the process of nitrogen fixation or which act as biofertilizers. Now the first type of organism or microorganism is rhizobium bacteria. Now rhizobium bacteria are rod shaped, then they are motile, that means they can move on their own. Then they are aerobic, that means they require oxygen for respiration. Then they are gram negative and non-spore forming, that means they do not produce spores. So these rhizobium bacteria carry the process of nitrogen fixation. Then rhizobium bacteria form a symbiotic association with the roots of leguminous plants. Now here we can see the root of a leguminous plant, and on this root number of spherical structures are developed, and these are called as root nodules. Inside these root nodules the bacteria multiply and live there, and here is the transfer section of a root nodule. Now here we can see this part is of the root. It shows presence of xylem, phloem. Now you know that xylem and phloem are conducting tissues. Then this portion represents the root nodule, and inside this root nodule the bacteria multiply and develop bacterial colonies in the cortical cell. So they develop a outgrowth on the root, here we can see, or a spherical structure on the root, and this spherical structure is called as a root nodule. Then these bacteria contain the genes, that is nod genes and NIF or NIF genes. Now nod genes are essential to identify the particular host. Host means to identify the roots of leguminous plants, and NIF genes help in the process of nitrogen fixation. That means nitrogen or the atmospheric nitrogen is converted into soluble form with the help of this NIF genes. Then in this association both the organisms are benefited, that is bacteria is benefited as it gets a place or a shelter to live inside the root and multiply there, and in return the root gets fixed nitrogen or soluble nitrogen, so that this nitrogen can be utilized by the plant for growth and development. Now bacteria or the rhizobia bacteria are specific to specific host. Now for example, the bacteria that is Rhizobium leguminosum is specific to the roots of pea plant, that means they will enter only in the root of pea plant and develop nodules. Then the bacteria that is Rhizobium fascioli is specific to bean root, that means this bacteria will form nodules only in bean plants. So these Rhizobia bacteria are host specific. So Rhizobia bacteria help to increase the fertility of the soil, as some amount of soluble nitrogen get mixed in the soil. So Rhizobia are the important bacteria which help in the process of nitrogen fixation. Now we will see the second type of microorganisms used as biofertilizers, and these are called as azotobacter. Now azotobacter are free living, then they are aerobic, that means they require oxygen for respiration, and are non-symbiotic nitrogen fixing bacteria, that means they do not form any symbiotic association between other organisms, and they are used as biofertilizers in the plants like rice, cotton and vegetables. Then the third type of microorganisms are azospirillum. Now azospirillum is free living bacteria, then it is aerobic and it is nitrogen fixing, or it carries the process of nitrogen fixation, and it is related to the roots of, or it is associated to the roots of corn, then wheat and jawar. So it can be used as a biofertilizer in crops like corn, then wheat and jawar. Then the fourth type of microorganisms which are used as biofertilizers are Anabina. Now Anabina is multicellular filamentous cyanobacteria, that means it is multicellular and it forms thread-like structures which are called as filaments, and cells are arranged end to end. Then Anabina may form symbiotic association with the roots of cycas, that is coralloid roots. Then this Anabina may form symbiotic association with the Anthocerous thallus. Now Anthocerous is a type of bryophyte. So Anabina forms association with Anthocerous thallus, or it may form association with coralloid roots of cycas. Then the Anabina consists of a specialized cell called as heterocyst. It is generally larger than other cells, and this heterocyst carries the process of nitrogen fixation. We have seen in our previous session in nostoc. Nostoc also shows presence of heterocyst. So these heterocysts help to convert atmospheric nitrogen into soluble form, that is carry the process of fixation of nitrogen. So Anabina can be used as biofertilizers in different types of crops. Now the last organisms used as biofertilizers are Azola. Now Azola is a free floating fern, that means it is aquatic fern and floats on the water, and it consists of a floating rhizome, and this rhizome shows presence of leaves and roots. Then the leaves show two lobes, dorsal lobe and ventral lobe. So in the dorsal lobe, Anabina filaments are present in the cavity, and the cavity is called as parenchyma, or the parenchyma tissue produces a cavity called as air cavity, and in this air cavity the Anabina filaments are present, and these Anabina filaments can carry the process of nitrogen fixation. Now here is the longitudinal section of Azola leaf. Here we can see the dorsal lobe, and inside this leaf a large air cavity is present, and in this cavity the filaments of Anabina are found, and here is the photosynthetic zone, that means here chloroplasts are present. So inside this cavity Anabina filaments are found, and these Anabina filaments carry the process of nitrogen fixation. and the azola can be used as fertilizer in rice field. So here we have completed the chapter number 11, that is enhancement of food production.